glycolysis, our pyruvate then makes its way into the mitochondria. It goes within the inner mitochondrial matrix, which is kind of like the cytosol of the mitochondria, that viscous fluid-like area within the membrane of the mitochondria. And within that area, the pyruvate that we created from glycolysis then enters what we call the linking step. So this is a small linking step that takes the pyruvate that we synthesized and actually creates acetyl-CoA, which is another enzyme involved here, or molecule, excuse me, involved here. So pyruvate comes in. Remember, we have 2 pyruvates, so we're only showing what happens to one of those. So everything that we have here, we're going to double it essentially. So for every pyruvate we enter, under the influence of the enzyme pyruvate dehydrogenase, as well as NAD+ that we insert here or invest here, we yield NADH carrying electrons as well, and we yield acetyl-CoA, which is going to make its way to the 3rd phase of glucose oxidation. And we also yield CO2. So this is the first time that we yield CO2 as a byproduct of this process. There was no CO2, there was no oxygen yielded or required in the first step in glycolysis. But here in the linking step, we do yield CO2, we do yield acetyl-CoA as the ultimate product, and then we do yield NADH carrying electrons to the mitochondria for oxidative phosphorylation as well, okay? And so going to the bottom of the screen here, we can look at the overall equation for this reaction. We've got pyruvate, which we invested from glycolysis. We've got CoA. We've also got the oxidized, form of NAD+, and then that yields us acetyl-CoA, CO2 as a byproduct, NADH, which is now reduced, carrying electrons towards the electron transport chain. So what happens to the acetyl-CoA that we then made at the end of that linking step? That makes its way to take part in the Krebs cycle. So the Krebs cycle I mentioned could also be called the citric acid cycle or the TCA cycle. This is a cyclical metabolic pathway. This happens within the mitochondria as well. It produces ATP. It is also going to produce some of our reduced carrier molecules that are carrying electrons as well. So far we saw that glycolysis produced some of those electron carrying molecules. The linking step produced some of those electron carrying molecules. And then here in the TCA cycle, we produce NADH+, 3 of them for each acetyl-CoA. Remember, this is happening twice because we initially produced 2 pyruvate. So everything that's happening here, we can kind of double it since we had 2 pyruvate, which created 2 acetyl-CoA. And so this step is going to happen twice per molecule of glucose. So that's 3 NADH+ in the reduced form. We also have another carrier that's introduced here, FAD, that is then reduced to carry electrons in the form FADH2, okay? We also produce 2 more CO2, again, as a byproduct here, and then we produce ATP as well, all right? I'm going to stress here again, the ATP that we've been producing so far has been by substrate level phosphorylation. So not as much ATP as we will see in the 3rd stage, and not free-floating phosphates either. These are phosphates that were attached to another phosphorylated molecule. Okay, so acetyl-CoA was our initial substrate. And then we produce ATP, the electrons that we mentioned, and then CO2. So bringing it down to our overall reaction here at the bottom of the screen, we invest acetyl-CoA, we invest the oxidized form of these carrier molecules, including FAD. We invest ADP, which is the unphosphorylated version here, and then we have to invest water. And that yields us CO2, our electron carrying molecules, which are NADH carrying 3 electrons, FADH2 as well as ATP on the back end. And because this is a cyclical reaction, we actually continue to yield CoA, and CoA will then go back and synthesize more acetyl-CoA from that linking step, okay? So here's a more detailed illustration of the Krebs cycle. Again, I'm not going to ask you to memorize these steps in any way, but I do want you to know what the substrates of this reaction are, what we invest, and what the products are, what we yield. Okay, let me pause here and see if there are any questions. So what questions do we have so far? So far, we talked about the overall picture of glucose oxidation. We talked about the first couple of steps, the first 2 stages, glycolysis, and the small linking step in between, and then the Krebs cycle, and how we take a molecule of glucose, and so far from that molecule of glucose, we've yielded 4 ATP molecules so far per glucose, which are coming from glycolysis and from the Krebs cycle. We've also yielded as a byproduct some CO2 as well. And then we've got a bunch of electrons that have been created that are being shuttled to the electron transport chain. Okay, so the third stage here is oxidative phosphorylation. This is going to require oxygen. This is where all of those electrons that we have been creating in the earlier stages of glucose oxidation, this is where those electrons are heading to. And we'll talk about what they actually do in a little bit. But the overall picture of oxidative phosphorylation is we move these electrons down the electron transport chain, kind of pass them from one complex to the other, and we synthesize the energy that's used to pass those electrons. So as we pass the electrons, we liberate a small amount of energy, and we take that energy in order to phosphorylate ADP with the inorganic phosphates that are in the mitochondria, okay? So in this third stage, we yield a lot more ATP. Okay, it's much more efficient. We yield a lot more ATP. And again, we are using inorganic phosphates and the energy that is liberated from passing electrons down the electron transport chain. So this is why it's crucial for us to be shuttling electrons from glycolysis to the mitochondria, from the linking step, from the Krebs cycle, because these electrons have a very important role in this third stage within the mitochondria. Okay. All right, so this is called the electron transport chain. We said it's a chain of molecules that are embedded within the inner mitochondrial membrane. The molecules go under oxidation to reduction reactions, going from high to low energy. So as they pass these electrons, they are releasing a little bit of energy as they go from the oxidized form to the reduced form. Remember, oxidation is loss, reduction is gain. And as we pass the electrons from one complex to the other, we take this energy and we use that energy to phosphorylate ADP and to make ATP via oxidative phosphorylation. Okay, the final complex in these chain of enzymes that are, these chain of molecules that are in the mitochondrial membrane, the final one is called ATP synthase. And so ATP synthase is the actual molecule that makes ATP. It's the final electron acceptor that is going to then phosphorylate that ADP. The last step in the process is for oxygen to accept the last electron. And that final accepting of oxygen, the final accepting of electrons by oxygen is what oxygen does. This is the whole reason that we breathe in oxygen, to act as the final electron acceptor in order for this ATP to be made. And this is why we say that this cannot happen in the absence of oxygen, because oxygen is required to grab those electrons from the final complex And then ATP synthase can then synthesize ATP after that final step is complete. All right. Okay, so we're kind of running short on time here, so I'm going to pause and see what questions have come up. We'll delve into this the next time we meet. We'll probably draw this out the next time we meet and kind of explain this a little bit better. This is probably the most important part of this process. This, I said, again, generates the most amount of ATP, and so it's going to be the most crucial step. It's also going to be the step that requires oxygen, and we'll talk about that when we meet next time. Okay,